So, and the story I want to tell you today is my first project at the IAS, which was on classifying prostate cancer, and it actually didn't start with cancer at all. It started here in the dining hall over lunch, and Arnie was talking to us. He came to lunch with us as always, and he was excited. He was like, guys, you have to read these papers that just came out in Nature back to back, and probably people here know about it. Those were 3 strong papers that were talking about inducing pluripotency in fibroblasts, and what they said was that p53 is a roadblock to that epigenetic reprogramming. And Arnie at the lunch table said, well, if that's happening in fibroblasts, that's happening in cancer. Gotta have a look at it. Hideaki was the first one to have a look at that in breast cancer, and then I took that on in prostate cancer. Now, here's another bunch of things that I probably don't need to talk about. What mattered at the time, 10 years ago, was the diversity of outcomes in prostate cancer. There's a lot of indolent disease, and then there's these 10-15% that die of the disease, right? And that's still a lot of people because it's a very common cancer. So that was a classification problem. Try to find these people. And our idea about it was very much alive by this hypothesis that, you know, when you have a quickly cycling cancer cell, you might actually start to epigenetically reprogram that cancer cell, and loss of p53 function might help both processes at the same time, and you would get something that is proliferating and that it's changing its lineage, right, and it's reprogramming to something aggressive. So how do we go about it? Well, we use gene expression signatures on transcriptomic data. We selected an array of gene expression signatures for embryonic stem cells, induced pluripotent stem cells, differentiated programs, and then genomic things. Obviously, p53 loss or gain, loss of function or normal function. And other common genetic alterations, P10, MYC in prostate cancer. And then I went to the literature and I read a lot of things about prostate cancer, and I came up with this list of things. In the end, you see like a proliferation signature, right? And I already put you together snippets of the data set that we looked at. And that's the class that we found that actually completely fits into the predicted picture. We have this red group here that has a stem-like features, dedifferentiated, loss of p53, loss of p10, gain of MYC, and highly proliferative. And so we turned this into a classification. This group, as hypothesized, had a very bad outcome. We did this in all the cohorts that were out at the time, and then published that. And yeah, while I was personally quite happy with this classification and the results, whenever I would go and show this to people, somebody in the audience would stand up and say, but that's really just proliferation, isn't it? And I did not like these comments very much, because we ended up doing a lot more work on cancer classification, and you start to learn that proliferation is a huge signal. It's a humongous signal. It's like almost like a third of all genes in the genome move up and down with proliferation. So you have this huge iceberg of a signal, and on top of it, you always see, your favorite proliferation genes like Ki67, TOP2A, RM2, whatever you want. It's always, those are the most significant ones. They're overwhelming, right? They're always there. And because of all our corrections that we always do, right, this is always what comes out on top. And my hypothesis is that, like, underneath, under the waterline, right, you have processes going on that you don't really see so much, but that might be important. And so for a while I looked at, epigenetic data and tried to get at this a little bit more. But it really didn't fully click until I moved to the Beatson, and I talked with people there, and the head of the screening facility came and said, like, well, if you have all these ideas, like, why don't you test them and do an siRNA screen? And so we did. So I ran back to my computer and started selecting genes for a screen. Meanwhile, a lot more data had come out, TCGA, sequencing data, so we ran our classification on all of these. Took as many cohorts as we could find, found a stem-like type, compared it to the most indolent subtype in each cohort, and looked at a bunch of significant genes across cohorts. And we came out with these like 230 genes, and it's full of cell cycle genes, obviously. And so then I went and I looked at every single one of them. And I subselected them by being annotated for the pathways I was interested in. So for P53, DNA damage response, like apoptosis, anything along those lines. And stemness, like embryonic things, early germline, oocytes, meiosis, spermatogenesis, anything around the package of early development, stemness, pluripotency. And we also included some of the proliferation genes because they were so striking, right? Tried to focus on more mechanistic things in the cell cycle. So we ended up with 48 hand-selected genes, and the screening facility tested them. And I just want to say about those, right? Those 48 genes themselves are very good predictors of outcome in any of the cohorts that we have today, right? So you see here survival, early recurrence. You see them coming back in castration-resistant prostate cancer, being high in metastatic prostate cancer, going up with Gleason score, distant metastasis, anything you want. And not only the average of those 48 genes works, but every single one of them works, because that's how they were selected. So they are all predictors of human outcome. And then we had our genes, we designed our screen. We took 3 commonly used prostate cancer cell lines, one of which, the link caps, is androgen sensitive, and the other 2 are androgen independent metastatic tumor lines. And then we went back to our hypothesis and we thought, okay, if we screen these genes and these cells, we should also remember that, like, if they really do make the cells more plastic, then they should also do contribute to treatment resistance. So we threw that in, and we took 2 of the most common treatments you can get in prostate cancer, which is docetaxel and enzalutamide, which is an androgen receptor inhibitor. So what I want to tell you today is about the docetaxel synergy, and I'm the one that's between you and lunch, so I think I have to make it short. So this is the guy that we found, the Elke. And the thing that was striking about it was that the siRNAs had a huge synergistic effect with docetaxel in only one of the cell lines, the link caps. And so who is this person? It's this large homolog-associated protein 5, and it's a microtubular bundler and microtubular organizer. It acts in mitosis and is actually essential in meiosis. You're infertile if you don't have it, because you can't go through meiosis. That's why it went in the list. Yeah, so here you can see it being expressed at the kinetochores. We checked it, everything is all right. Like, indeed, you put it together with docetaxel, the cells die, the siRNAs are working. Question is, it makes sense that a microtubular bundling gene should interact with docetaxel, which is a microtubular targeting agent. But why only in one of the cells? So is it androgen-dependent effect? And it is, so this data, like down here in C, you see the dose response curves, and in blue, that's the Elke knockdown, dose response to docetaxel. And when you look at the yellow line, you see that that's when you additionally knock out the androgen receptor in the cells. So you ameliorate the effect. And then when you take the same cells and you desensitize them to androgen, the effect vanishes. No more symmetry, no more synergy. Right? And then when you use an androgen inhibitor at the same time as docetaxel, nothing happens. You still get the synergistic effect. So you get a graded response depending on how much the cells have adjusted to the androgen setting. If you're in an androgen-independent cell that's fine without it, you have no effect. If you're an androgen-sensitive cell, you have a big effect. And we investigated this a little bit further. It's not a direct interaction, and it's definitely something that stops the cells in metaphase. So we believe that it's the central spindle part that breaks the cells, right? One explanation that we came up with is that it's like a sort of a perfect storm. We know from the literature that androgen generally suppresses tubulin in the cells already in interphase. The LGAP5 sprouts some of the central spindle microtubula, so you would lose that. And then docetaxel would take care of the rest at a very low dose. So we thought this was really interesting. I think it's really cool that we started out with this like stem-like group. We looked at these like stemness-related things. We found this meiotic organizer, right? And it leads us to basically the androgen receptor system, which is, in the end, the developmental pathway of the prostate, the most early driver of the development of the organ. We have our molecular classification. We can robustly detect the subtype. And we see that when we go in and we actually look sort of in the middle of the iceberg for these things, we might find novel regulatory things. This is generally what I'm interested in. I'm now interested in hormone-driven growth across cancer, along with all the other classification problems that I like to play with. So, and with that, I want to thank everyone at the Bietzen Institute who helped me put together the data. And of course, there's a glaring omission. Thank you, Arnie. Thank you. Without you, I wouldn't be here. And I wouldn't have been in many other brilliant places. Arnie typically exposes you to all sorts of wonderful culture, wonderful people, wonderful meetings, and wonderful science. Happy birthday.